Our next speaker is Darylyn Mahoney, a pediatric genetic counselor at Children's National Hospital. Darylyn works closely with children and families navigating rare genetic diagnoses, including LCC, and brings both clinical expertise and a deep understanding of the questions families face. Today, she'll help us better understand the genetics of LCC and what they mean for patients and families. Please join me in welcoming Darylyn.
Good morning, everyone. Thank you, Ashley, for that wonderful introduction. And then thank you, Dr. Fraser, for that wonderful talk. Again, my name is Darylyn. I'm a genetic counselor, and I am going to talk to you guys about the genetics of LCC today. You're going to hear a lot of things today and over this weekend about the genetics of LCC. So what you're going to do with me are learn all the things that are absolutely positively necessary to understand all of the talks that are going to follow me. There are going to be plenty of brilliant people that are going to be talking about the nitty-gritty details. So I'm going to go over the surface to make sure that you're getting the major takeaways about the genetics. All right, so I have no disclosures and no conflicts of interest, so this is the easiest slide. And so then we're going to talk about our objectives today. So there are three major things that I want to make sure that we cover. So the first is to understand the genetic cause of LCC. We're also going to learn how SNORD118 variants lead to LCC. And again, this is variants because we are people and not mutants. So we're only going to hear variants from me. And then we're also going to discuss inheritance and implications for families that have variants in SNORD118. All right, so a LCC clinical overview. This is probably not something you guys need. I'm sure you know just as much as I do at this point. So again, just in the general sense, you can't talk about LCC without talking about that classic clinical triad. So again, we have that leukoencephalopathy, which are those white matter changes, the calcifications, and the cysts. And because I love arrows that show me what's on imaging, I also gave you guys arrows that shows what's on the imaging. And so anything that's denoted with that single eye is going to be those white matter changes, that white matter involvement, or the leukoencephalopathy. Anything with the double eye is going to be the calcifications, and anything with that triple eye is going to be the cysts. And so we'll see the cystic degeneration and also those irregular calcifications in that 2 and 3. All right, and some additional features that we'll also see include seizures, spasticity, or those movement difficulties. There can be some cognitive differences and developmental delays. And we also know that age of onset and disease progression are highly variable amongst each patient that has LCC. There are also additional features that we're consistently learning about. And as we just continue to learn more about LCC as a condition, this is going to get a little bit longer as we understand it more and more. And there's a few things that I like to reemphasize. There's going to be a lot of reimposition. And one of the things that I like to emphasize is that every single patient is different. And so I like to say that everyone can present a little bit differently. And so in that way, we're all just like beautiful, unique snowflake. All right. So as Dr. Fraser delved into before, I want to make sure that I just go back to this little history lesson. So in 1996, which I personally refuse to believe was 30 years ago, deeply refuse to believe. French researcher Dr. Philippe Lebrun, our initial namesake, and his colleagues were the first to describe that condition. And so they had 3 unrelated patients with that classic clinical triad. And so those patients had the clinical triad. They also had slowed cognitive performance and seizures. And those 3 patients had a difference in the age of onset. So that ranged from early infancy to adolescence. And then 20 years later, now 10 years ago, in 2016, another set of researchers were able to identify the SNORD118 gene and found that was the genetic cause of LCC. And so in that study, they had 40 patients from 33 individual families that all had that classic LCC triad. And so they found that those patients had a clinical onset that ranged from infancy to about 54 years. So again, going back to those snowflakes is just such a significant range in how everyone can be affected. And so they actually did exome sequencing first, which is something that you guys might also be familiar with. And they weren't able to identify anything with that exome sequencing. And while we love exome, it is fantastic at picking up variants that are in genes that code for proteins. This was an issue because SNORD118 is a non-coding gene. And so they ended up doing genome and then linkage studies. And with that, they were able to identify the SNORD118 gene. And they found that every single affected patient had two SNORD118 variants. And so of those 40 patients, they were actually able to identify 36 unique novel variants within this cohort. All right, so now we're going to delve into understanding SNORD118, what it does, and why it's so important. So SNORD118 is incredibly important because it encodes a UAA, or a small nucleolar RNA. And so SNORD118 helps to regulate ribosome biogenesis, so the production of ribosomes. And as Dr. Fraser mentioned before, I know how the light works too, so that's fantastic. It's really important for this top part of the ribosome. And we're going to talk about, this is something we're also going to reemphasize. The ribosomes are so incredibly important because the ribosomes are what creates the proteins in our body, especially proteins that we can't get from steak and chicken. These proteins are incredibly important. They do all the jobs that help us function as humans. And so what this UH-SNO RNA does is it also helps with ribosomal RNA processing. So without that ribosomal RNA, we can't get this top part of the ribosome. So we can't make these ribosomes. And again, without those ribosomes, we do not have the proteins that we need. And we need those proteins for normal cellular function. So if we don't have a SNORD118 that works, we can't get that ribosomal RNA processed. We can't get that top part of the ribosome. So we can't get those proteins, and puts a lot of stress on our cells and on our body. So then how do variants in SNORD118 cause LCC? So what we know are that variants in SNORD118 can essentially break the gene and make it very hard for it to do its job, and pretty much decrease or eliminate its ability to do its job at all. And so when SNORD118 is broken, that UH-SNO RNA cannot be properly made. Ribosome formation is impaired. There's a lot of cellular stress, and so cells will want to stop dividing, stop growing in different tissues and organs. And then there's two things that we'll see that Dr. Fraser also touched on, and that's that small vessel dysfunction and also progressive microangiopathy. And so that's small blood cell damage. And with the dysfunction, that just means the tiniest blood cells in our bodies aren't able to deliver blood and oxygen in the way that they are normally expected to. And so that is responsible for the cognitive and a lot of the neurological things that we see with LCC. And again, that microangiopathy, responsible for that classic clinical LCC triad. So just in case I lost you anywhere, I also have another summary. And so biggest takeaway, too long, didn't listen. We have our SNORD118 gene. If that SNORD118 gene doesn't work, we don't get that UH-SNO RNA. If that UH-SNO RNA doesn't work, then we don't get the ribosomes. No ribosomes, no proteins. And proteins are the building blocks of life. And I want us to say that together because that's super important. And I like to have a teach back method. So on the count of three, we're going to say that one together. 1, 2, 3. Proteins are the building blocks of life. Thank you guys very much. All right, so the major takeaway is no SNORD118. We're going to have that impaired protein synthesis, and therefore our body does not want to do what we want it to do. All right, so we're going to touch briefly on inheritance and recurrence because I'm going to touch on this again a little bit later if you join me later when we talk about some family planning options. So some major things to think about with inheritance of LCC, we all know that it is an autosomal recessive condition. And so with autosomal recessive, we have one copy of SNORD118 from one parent, one copy from the other. And we know that you have to have those genetic variants, those genetic changes on each copy so that there is no longer a functioning copy in order to have the symptoms of LCC. Now, as a parent, maybe as a sibling, you might have only one variant making you a carrier, but you still have a functional copy of SNORD118. So you are lucky enough to not have any of those symptoms, hopefully. And so some considerations for recurrence risk for carrier parents, they have a additional 25% risk of having an affected child with LCC in the future, a 50% risk of having a child who is a carrier like yourself with just the one variant. And then there is also the 25% chance of having a unaffected and non-carrier child. All right. And so some Some key takeaways from our time together. LCC is caused by biallelic 2 variants, 1 on each copy, in the SNORD118 gene. SNORD118 encodes that small RNA that is involved in creating those ribosomes. Ribosomes make those proteins. And we know a lot, but we don't know everything, and research is incredibly important. So it is wonderful that you guys are either joining us here today in person or virtually, because collaboration between families, clinicians, and researchers is so important and so essential for future therapies. And I always like to throw my references in there just in case. But thank you guys for your time, and I'm happy to take any questions, and if there's any I can't answer, I will gladly punt them over to Dr. Fraser. Thank you.